area of pharmacoeconomics and the development of nanocarrier diagnostic in the management of cancer by Dr. Anup and by Dr. Sharad Prakash Pandey. Today, we have board with us a distinguished scientist and academician, Dr. Gautam Rath, Professor, Department of Pharmaceutics, Shiksha O Anusandhan, deemed to be University, Bhubaneswar. I request Dr. Neeraj Mishra to introduce our speaker to the gathering. Sir, please.
It gives me immense pleasure to introduce our first speaker of today's session, Dr. Gautam Rath. Dr. Rath is a professor, Department of Pharmaceutics, Shiksha O Anusandhan, deemed to University, Bhubaneswar. He is a leading scientist in the field of cutting-edge platform technology, that is nanotechnology, for site-specific targeted delivery of drugs and bioactives. Recently, Dr. Rath has recognized as the world top 2 scientists published by Stanford University, USA in the year of 2019, 20 and 2021. He is also the recipient of top 10 scientists of the India by Career 360 in the year of 2018. He is a member of many professional and national and international societies. He serves as a various expert and reviewer panel in the medical biotechnology and nanotechnology. He has been responsible for many research projects worth more than 10 crores funded by the government organization DVT, DST, AICT, ICMR. He has published more than 150 scientific articles in recognized national and international journals, and he has authored 4 textbooks in the field of pharmacy. He's having the citation more than 6,000, H-index 47, I-index 108. The innovative character of his research activity is also reflected by 25 patents out of the 6 patent technology transfer to government, national agency, and reputed company. He has published more than 20 book chapter and more than 60 presentation at scientific conference, and he has supervised 56 MPharm and guiding 4 PhD. Okay. And now I invite Dr. Gautam Rath to take over the session. Sir, please. Thank you.
Thank you, Dr. Neeraj, and all the participants.
Sir, your voice is a little bit low.
Neeraj, am I audible?
Sir, voice is coming slow.
Am I audible?
Yes.
Am I audible?
No, sir. It is not clear.
Sir, audio is very low, sir. Can you, sir, please increase it? No, sir. Audio is very low, sir. Can you, sir, please increase mic volume, sir?
Am I audible?
Sir, audio is very low, sir.
So very low, sir. Your voice is very low. Like in practice session, it is very clear.
Our speaker is going to join soon. Please be patient.
So is it audible now?
Sir,
Audible?
sir, again, little bit slow.
Slow. Okay.
Sir, if you able to remove that
So is it
headphone and
audible
then
now?
try to speak, sir.
Is it audible now?
Yes, sir. Please share
It-
your screen, sir.
It is clear. So my voice is clear.
Yes, sir. Yes.
Okay, okay, okay.
Sir, your presentation is also visible, sir. Sir, are you able to listen me, sir?
Right. Today email.
Uh,
Hello. Am I audible now?
Yes, sir. Very clear, sir.
Okay, okay.
Audible, sir.
Okay. Dr. Neeraj, thanks. And it is really a difficult time. We are facing some problems with the connections. So I'm
Sir,
extremely
again, it
sorry
become
for
low,
that.
sir. Voice, please, sir.
So is it clear?
Yes, sir.
Okay.
Sir, please give the presentation full screen, sir.
Okay, okay, okay.
Yes.
Is it fine? Hello.
Sir, kindly present the screen in full screen, sir.
So it is already in full screen
Sir?
mode.
Gautam, sir.
Yes.
Is it audible to you, sir?
Yes, yes. It is audible and it is already in full screen mode. Hello.
Sir? Sir Gautam, sir?
Yes, sir. Yes, sir.
Sir, kindly put in a full screen, sir.
So it is already in full screen.
So
Sir, it's not in full screen.
voice is not audible, sir.
Boss, it is not audible.
Yes,
Is
sir.
it clear?
Sir, okay, sir. You can start, sir. You start with the full screen, sir. Sharing by full screen, sir.
Yes, sir. Now it's full screen, sir.
Oh, it is okay, sir.
Okay.
Yeah, okay, sir.
Okay. Okay. Fine. Okay. So I'm extremely sorry due to some problems with the connections. So I'm audible to all of you.
You can start, sir. You are audible to all.
Okay. Okay.
Thank you.
Okay. So first, I would like to thank the organizing committee, and actually it is very nice to see Dr. Neeraj after a long time. So we are actually friends from 2001, and it is really thanks to Dr. Neeraj and thanks to the organizing committee for giving me the platform to share on the topics that is the role of NDDS in cancer. And dear participant, we have discussed what is the role of NDDS in cancer. But first, what is the relevance of this topic? See, in case of cancer, although there are different methods are available today, radiotherapy, chemotherapy, or surgical therapy, or immunotherapy, but none of the method, actually none of the treatment protocol ensure a complete therapy or percent success rate in case of cancer, it is difficult. Even if whatever the technique that is used for the treatment of cancer, chemotherapy play an integral role. Even after surgical therapy, the patient need chemotherapy cycle at least for 5 to 6 cycles of chemotherapy is required for the patient undergoing surgical therapy. But whenever we discussed about cancer, the word cancer, it is a little bit scary. But cancer also provides a lot of opportunity for the scientific community. And because of the contributions of the scientists in cancer, the molecular mechanism of the cancer, it is explored to a certain extent. And see, actually, we want the human beings.
Hello, Sir Gautam, sir.
Yes.
Your voice is very low. You please speak a bit louder. We will try again.
So, am I audible?
While you are saying, am I audible? It is clear. After that, you are going down.
Okay,
You have to
okay.
keep the same temper throughout the presentation. Then only
Okay.
no audience can listen you.
Okay.
You
So
just
it is
keep
audible
talking
now.
loudly throughout your presentation. Keep talking
is-
loudly throughout your presentation.
It is clear?
Now it is clear, but you have to maintain that.
Okay, okay, okay.
Thank you, sir. Thank
Fine, fine, fine. So whenever we're talking about the cancer, basically the human beings want to be immortal. Through our research effort, actually we want that is there any possibility or is there any mechanism we can identify through which we can become immortal? But it is the cancer. It is a natural process from where scientists can identify, yes, a cell, a living cell, may be immortal. And and how the cell becomes immortal, there are certain genetic changes and there are certain genetic mechanisms. Although the scientists identify some of the genetic attribute that makes the cell immortal. See, this immortality of the cancer, it is a challenge for the scientist to identify a mechanism to make the cell mortal. And it is the immortality character of the cancer that helps the scientist to identify a new drug. For example, today, the most effective drug which are selected in the later stage of cancer are monoclonal antibody. So is it possible? Is it possible to think about the monoclonal antibody without cancer? Because in the preparations of monoclonal antibody, we need a myeloma cell, a cancerous cells. See, although cancer is a challenge, But there are some character due to which the cells are used today for some other purpose. But when we are talking about the topic of the seminar, so NDDs, novel drug delivery systems in cancer, that means we are discussed on the chemotherapy. So why not the conventional mode of drug delivery systems are useful in cancer? Why there is a requirement of a new drug delivery systems for the treatment of cancer? The reason, see, just look at the slide. The drugs which are developed against cancer, they are programmed to kill those cells which can replicate in a rapid space, which can proliferate in a rapid space. That is the reason why the anti-cancer drugs are, if they are not selected or if they are not targeted, so they can kill all those cells which can multiply in a rapid space. For example, the cells which are multiplied in a rapid space in our systems, first is RBC, then skin, then hair cell, then the bioflora, then the mucosal cell. So when we look at what are the side effects of cancer, definitely the major side effect would be anemia, because the cancer cell can kill RBC. So RBC, why RBC? Because RBC multiply at a rapid space, and the drug are designed to kill all those cells which can multiply in a rapid space. Exactly the GIT disturbance. Why GIT disturbance? Because GIT, the mucosal lining and the epithelial lining, it is replenished after 21 days. That means the life of the mucosal lining of GIT, it is 21 days. After 21 days, the new cells replaced the old cells. That means the epithelial cell of GIT multiply in a rapid space, and anti-cancer drugs can kill those cells, and that result the GIT disturbance. Exactly the bioflora, the bacteria can grow in a rapid space, and the anti-cancer drugs can affect the growth or the density or the populations or the biodiversity of the bioflora, which cause a number of GIT problems. So this is why there is a necessity to rethink the drug delivery approach for anti-cancer drugs. Because whenever the anti-cancer drugs are administered through conventional like tablet. So the drug, it is absorbed and distributed throughout the systems, irrespective where the cancer cell is present. It is distributed throughout the systems. So when the drug reaches the off target other than the cancer cells, there they can produce the side effect. So what is then how the drug, anti-cancer drug, are localized? How we can make target, how we can make, or how we can design a approach that ensure maximum drug concentration near the cancer. In order to target environment of the cancer cells, how the cancer cell behave, what is the anatomical locations of the cancer cells. Because without these informations, we cannot prepare a appropriate, or we cannot design a approach that helps in the localizations of the drug near the cancer cells. So when we discussed about NDDS, what is NDDS? Means there are conventional and there are NDDS. So any drug delivery systems which are recently launched or recently designed and would be defined as NDDS, novel drug delivery systems. So it is not that tablet are conventional. We can make the tablet NDDS. So it depends the tablet which are available in market. What is the drug delivery mechanism? So if some changes in tablet which makes the tablet program delivery, which makes the tablet to deliver the drug in a order we want, that makes a novel drug. It is not that the liposomes, nanoparticles, carbon nanotubes, quantum dot are novel drug delivery systems. Any changes in drug delivery systems, any added feature to the drug delivery systems, which makes that system more effective than its previous, then we may call it a novel drug delivery systems. But before that, we should identify the objective. Why we think for a new drug delivery systems? What are the objectives? See, there are the major aspect to design a drug delivery systems to make the drug localized near the cancer cells, so that the dose that is required may be reduced and dose dependent toxicity may be reduced, and the therapeutic potential of the drug may be further enhanced due to accumulations of the drug near the cancer cells. So these are the objectives. So just imagine there is a blood cancer and there is a cervical cancer. In case of blood cancer, the microenvironment, it is completely different from the microenvironment if you compare with cervical cancers. So accordingly, as the microenvironment, it is completely different from one cancer to another cancers. So the drug delivery approach, the design, the carrier system has to be changed, isn't it? But apart from that, that is not the only objective, although the major objective to localize it. But that is not the only objective, because what are the challenges? Challenges, the solubility of drug. Challenges, the permeability of drug. Challenges, the stability of the drug. Challenges, the dose of the drug. These are the pharmaceutical challenges. And after that, there are other challenges. How we can confine, how we can restrict the localizations. So there are a number of challenges are there with anti-cancer drug. So when we select a novel drug delivery systems, we should identify what are the key challenges, what are the key problems, and how those aspects can be overcome. So in that respect, we have selected the carrier systems in front of you, that is nanofiber. Nanofiber, it is also it is actually what is nanofiber. Definitely it is a platform, it is a carrier, may be explored for anti-cancer drug delivery systems. But why? Why one can select nanofiber? See, nanofiber, it is just like a paper. So all of you see the paper, isn't it? So the nanofiber also, it is almost similar to a paper, but in paper, the material that is used for paper, it is cellulose. So cellulose fiber actually are arranged, and ultimately we have a sheet of cellulose fiber. Basically, they are arranged to form a paper-like structures. But the fiber that is used to make paper, the diameter of the fiber, it is approximately 10 to 15 micrometer to 500 micrometer. Normally, whether it is a Whatman paper or it is a blotting paper or it is a just paper of books or whatever the paper. Usually the fiber, cellulose fiber, which are selected, the diameter it is from 10 or 15 micrometer to 500 micrometer. So just imagine if someone selects a fiber of 15 nanometer to 200 nanometer. That is, if you convert in micrometer, that is from 0.015 micrometer to 0. Then the final product, that a result after the arrangement of those fiber, it is called nanofiber. So it is a first participant. Actually, they are very curious. Anything that is exist in nano structure, is it visible to naked eye? So see, nanomaterial, it is classified in 3D, 2D, and 1D. So if you just take example of any nanoparticle. So the nanoparticle, it is a 3D nanomaterial that mean in all directions. The particle exists in nanoscale. But in case of nanofiber, it is a 1D nanomaterial. That means it provides a nanometric feature only in one direction, one directional nanometric feature. Therefore, this nanofiber, it is visible to naked eye. That is one of the most interesting aspect of nanofiber, because once it is visible, we can see it, and there are excitement, and we can feel, we can analyze the stability through the naked eye. So although we need sophisticated analytical tool for the analysis of nanofibers, but it's just like a paper. The final product, that is the final nanofiber, it is just look like a paper, and that paper consists of material which are 1D nanomaterial, 1D. And actually the history of nanofiber, it is not, it is very recent, because in 1908, the concept of nanofiber is launched by one scientist But most the most striking aspect of nanofiber, it is only in 2000, just 22 years back, scientists realized the potential of nanofiber, and there are a lot of applications it is investigated on nanofiber. Only that what I like to convey, just 20 years back, the scientists investigated the potential of nanofiber. And see, in case of liposomes, it is almost 50 to 60 years back. The liposome is punched, and we have today 3 to 4 liposomal product, specifically in doxorubicins and amphotericins B, and for some antiviral drugs, it is approved by FDA, which are liposomal products. But just the age of nanofiber, actual age of nanofiber, is 20 years. And since last 20 years, there are a number of products available in market that are prepared from nanofiber. Why nanofiber are so popular? Because it is a stable product. Ultimately, it is stable. And the stability of nanofiber, it depends on the polymer that is selected. For example, in case of paper, the polymer that is selected, it is cellulose. But we can prepare nanofiber from large number of polymers. From a large number of polymers. Any polymer you think, you can select that polymers to prepare the nanofiber. Why I am actually and highlight on the polymers? Because it is the polymer that makes the nanofiber unique. It is the polymer. See, today, the nanofiber, it is extensively used to make filter paper. And I think there are 20 to 25 masks is developed and patented by Israel during COVID period, and are more effective than N99. The masks which are prepared from nanofiber, and just it is from last 2 to 3 years. The development, it is from last 2 to 3 years. And the masks which are developed from nanofiber are much more effective than N99 mask. So why nanofiber are excited materials for masks? See, as in N95, I think you may have experienced there are little suffocations when N95 mask is put. Suffocations. Why it is suffocations? Because suffocations that directly indicate the pores, the pore density which are available on the mask, pore density. So we cannot make a fiber with a very large number of pores, because there is no significance to take a mask with a large number of pores, because the objective of that mask to protect from virus, and the virus are usually, it is less than 0.1 micrometer, usually. So we need the pores which are less than 0.1 micrometer, less than 100 nanometer. So just imagine, we must have used the membrane filter, and the pore
Sir?
diameter of membrane filter.
Sorry for interruption, sir. Please
Yes.
sir, move the slide, sir.
Okay.
Slide is
Just-
not moving, sir. Yeah, please.
Okay. So the membrane filter, what we are using.
So the
That is-
slide is about systemic side effect of chemotherapy. Sir, please move the slide, sir.
Okay.
Presentation slide, sir.
Is it clear?
Yes, sir.
Okay. Okay. So in case of membrane filter, it is
Sir,
0.
sir, once more time, I am sorry to interrupt you, sir. So the
Yes.
conclusion time is by 11:30, sir. Please try to conclude within that time, sir.
Okay.
Yeah, thank you, sir.
Okay.
Please continue, sir.
Okay, fine. So what we have discussed, that membrane filter, just imagine the pore diameter, it is 0.2, but when someone uses the membrane filters, it is very difficult to filter. We need a vacuum. So just imagine if we are selecting a mask with pore diameter that is less than 0.2. So how difficult to breathe. But in case of nanofiber, it is possible because it has the number of pores. The pore density is much more. So in case of a normal paper, the pore density is usually 30 to 40%. But just imagine if we have a paper that is consists of nanofiber, the pore density is more than 90%. See the density of pores. So that pore density that helps in the breathing. Therefore, nanofiber, it is extensively used to make the mask. So why that nanofiber, it is selected for anti-cancer drug delivery systems, for anti-cancer drug delivery? See, there are a number of anti-cancer drugs which are poorly soluble. So nanofiber has the potentials to improve the solubility of the anti-cancer drugs. Nanofiber has the potential to make the drug soluble, more soluble, because during the manufacturing, there is an instant solidifications of the liquid jet. When there is an instant solidifications of liquid jet, there is a possibility the drug may change its physical state. That means a crystalline drug may transform into amorphous state. And we all know that amorphous state of the drug, it is much more soluble than its crystalline counterpart. Nanofiber, it has a very high surface area. So the high surface area makes the nanofibers mucoadhesives. For example, there is a cancers on mucosal cell lines. So we require that nanofibers should retain on the mucosal cancers. So since the nanofibers has a high surface area, that helps the nanofibers to retain on the target site. Nanofiber, since it is a solid carrier systems, and the steps, the unit operation steps, the operational steps that requires to prepare the nanofibers, it is a single steps. So it makes easy for all of us to scale up, which is very, very difficult in other nanomaterials. And the biodegradability, it is not a property that is directly related to nanofiber. It is related to the property of the polymers. Which polymer we are selecting to prepare the nanofiber? Usually, since the nanofibers, if it is used for drug delivery aspect, definitely we have to select those polymers which are biodegradable in nature. This is just a set of electrospinning setups. Okay, the nanofiber can be prepared by a number of techniques, but electrospinning that is most commonly adopted to prepare the nanofibers. And this work on a principle of two things, only two things. That is the conductivity of the material and the surface tensions of the polymer substance. Actually, we have applied a conductivity that should overcome the surface tensions of the polymer substance so that the polymer substance is stretched to prepare the nanofibers. That is the basic principle. The two aspect that is essential in nanofibers, the conductivity of polymers and the surface tensions of the polymer substance. These two things must be optimized because the conductivity of polymers, it depends on the solvent that is selected to make the polymer substance. And and in electrospinnings, we have features to apply a voltage of more than 50 kilovolt. Just imagine in high tensions where the voltage in high tensions where that is 15 kV to 20 kV. But in electrospinnings, there is provisions to apply up to 50 kV of voltage because we need high voltage so that applied current should overcome the surface tensions. And when there is a movement, the conductivity overcome the surface tensions of the polymer solutions, the polymer solution automatically stretch and collected on the collectors. So the stretching of polymers that helps to make the polymer into a fiber of diameter less than 200 nanometers. These are some of the applications of nanofiber. It is already discussed, and there are a number of products available on nanofibers today. There are some of the product that is available in nanofibers, and I just want to highlight the first one. See, the face mask that is developed by eSpin Nanotech. eSpin Nanotech is the only manufacturer of electrospinnings in India, and they have developed this face mask during the COVID, and it is very, very effective. Even it is more effective than N99s. And just see what are other products which are available in nanofibers that indicates the potential of nanofibers for biomedical applications and for other applications. I want to just highlight some of our research outcomes that is based on nanofibers, and here actually we have developed a nanofiber that is the polymer selected, that is PCL and chitosan. Okay, and we have prepared the nanofibers using the drug cisplatins. The objective to select the cisplatins, we need to improve the solubility of cisplatins, first objective, and the second objective, this particular study, that is it is intended for cervical cancers. So in cervical cancers, since it is actually we want a mucoadhesive drug delivery systems that can help to retain near the cervical cancers. And second objective, is it possible to improve the solubility of cisplatins by taking the advantage of electrospinning techniques to make nanofibers. And these are some of the photographs of our nanofibers. And see, if someone want to analyze or want to see the effect of the fabrication techniques on the physical state of the drug. Because first, in order to assess the solubility, we have a number of techniques, but X-ray diffractions, TGA, and DSC are most commonly adapted techniques to analyze the change in physical state of the drugs. So here it is X-ray refractograms. So see, in X-ray, in first and second, the first image it is in red and the second image in blue. See, there is the drug is present and there are some broadening. The X-ray diffractogram, there is a broadening, slight broadening. And that slight broadening of peak, it indicates some sort of physical changes of the drug in the nanofiber takes place. And these are the release study. Since the polymer that is selected, it is PCL and chitosans. See, chitosans to make the nanofiber mucoadhesives and PCL to control the release of the drugs. Okay, and almost 70 to 75% of drug is released after 30 days. 30 days. And PCL, it is very slow biodegradable polymers. So the implant which are available in market for anti-fertility hormones, that implant are also the polymer that is selected for to prepare that implant is PCL. So PCL, due to the presence of that PCLs, we have a prolonged release profile of the drugs. And this is the swelling, because swelling that affect the drug release from the fibers. This is the tensile strength, because if we are making some paper-like structure, and that is meant for a intravaginal applications. So it should have a minimum physical and mechanical strength. So in order to ensure what is the mechanical strength of our nanofibers, so we have tensile strength, and we got very good mechanical strength of nanofibers. Just if I compare that mechanical strength of nanofibers with a normal fiber, it is almost 10 times. 10 times the mechanical strength of nanofiber, it is 10 times more in compared to normal fibers. That is because of the fiber density. जैसे हम सभी को पता है, कुछ छोटे-छोटे पेपर्स, छोटे-छोटे अगर लकड़ी को लेके एक साथ अगर उसको पकड़ा जाए तो उसका मैकेनिकल स्ट्रेंथ इट इज मच मोर if you compare with a single. Similarly, in case of nanofibers, per unit centimeter or per unit millimeter, the number of fiber it is much higher if it is compared with the normal fibers, and that is the reasons why the mechanical strength of nanofiber is much higher. So this is the permeability studies. This is the cell viability studies. This is the tumor regressions. Actually, we have induced the tumors in animals by xenograft models and see the changes in nanofibers. This is the plain drug. If you compare the tumor regressions potentials of plain drugs and just see the potentials of nanofiber, that is only because the nanofiber it retain at the specific site, and that makes the drug more more effective, and it is the drug it is since the concentration of drug it maintains to a IC50 levels near the cancers, that helps to replace the tumors. This is the histopathology. It is very essential because the nanofiber, the nanofiber that is designed to retain to a specific site, and the chemotherapy drugs are known to produce a side effect on the epithelial cells. So it is a histopathology structures. So
Sir,
in histopathology, yes.
sir, please bound this.
Okay,
Bind
just
this-
5 minutes, just 5 minutes, just 5 minutes.
Yeah, sure, sir. Thank you. It is time bound session, sir.
Okay, thank you.
Thank you, sir.
So in histopathology, we didn't have any abnormality in histopathology structures. And we have a second study where we have selected those taxes for buccal cancers. And here also, why it is buccal cancers? Because again, we want to deliver the drug to site specifics. Here also, we have selected a mucoadhesive materials and make that structures adhesive to the buccal cancer site. And, it helps to retain at the site, and it maintain a effective drug concentrations over a long period of times. And this is the results. So these are some of the other applications of nanofibers that is developed by our research groups. We have a number of drugs selected for nanofibers. So we can, you also, if some of you want to develop nanofibers or want to discuss more on the nanofibers, then you are welcome to discuss with me. So these are some of the other studies of our research groups. So with this, thank you very much for giving me this opportunity to discuss. Okay, any questions?
Thank you so much, sir. You bestowed us with your wonderful knowledge on the topic of nanofiber-based drug delivery platform for cancer treatment. We would like to give you virtual felicitation to our guest speaker, Dr. Gautam Rath, for sharing his expertise in the session. Sir, please accept our felicitation, sir. Thank you, sir.
Okay, thank you.
Ma'am, slide is not viewing.
Now the time to proceed to the next session. I would like to call Ms. Ankita Kishore to introduce our next guest speaker to the
Okay,
gathering.
should I leave now? Should I leave?
Sir?
Should I leave?
Yeah, sir. Yeah, sure, sir. Thank you, sir.
We have an eminent academician and researcher as a resource person for the today's session. He is Dr. Natarajan Jawahar, Faculty,
Ma'am,
Department
slide
of
is not moving.
JSS University, Ooty. It's my pleasure to introduce Dr. Natarajan Jawahar to the virtual gathering. Sir has earned his B Pharm from Arulmigu Kalasalingam College of Pharmacy, M Pharm from KM College of Pharmacy, and PhD. It is okay. And PhD from JSS University, Mysore. His 20 years experience includes both teaching and research. Sir has received several awards, including Career Award for Young Teachers, Tony B Award, Academic Travel Award, Best Industrial Pharmacy Poster Award, Best Presentation Award, Young Scientist Fellowship 2017, Young Talent Award 2018, and Best Teacher Award 2022. Sir has also received travel grants from various funding agencies such as DST, CSIR, ICMR, and FIP for attending various conferences. Sir has published 86 articles in peer-reviewed index journal with an impact factor ranging between 1 to 7.5 and presented 30 research papers in national and international conferences. To his credit, he has been granted 1 patent and 5 filed and published patents, and has also filed 3 Indian patents. He has also authored 2 books by Lambert Academic Publishing, Germany, and 6 book chapters for Apple Press, USA, CRS Publication, and Reverse Publishers, UK. Sir also has been involved as a resource person in national level seminars and delivered 20 lectures in various universities across Tamil Nadu. He has guided 53 PG students, 1 PhD scholar, and 3 PhDs from ICMR. His current area of research is formulation and in vivo evaluation of lipid-based novel drug delivery system for improving the bioavailability of drugs targeting site-specific tissues and organs. Sir is also having lifetime membership.
एलडब्ल्यूपी लगेगी, इसकी छुट्टी तो चली गई है, इसकी सीएल थोड़ी मिलेगी, या इसको एक महीने तक कोई सीएल नहीं मिलती इसकी रश्मि की.
हेलो राम सर, हेलो राम सर, हेलो सर, आवाज आ रही है.
Sir is also having lifetime memberships of various professional bodies like IPA, APTI, APP, AAPS, and SPDS, and also recognized as a reviewer and editorial board member of various international and national index journals. Now, I request Dr. Natarajan Jawahar to take over the session. Sir, please.
Thank you, madam, for your nice introduction. Hope my voice is audible.
Yes, sir, it is audible. So you can share your screen.
Yeah, definitely. Hope the slide is shared.
Yes, sir, it is visible.
So, good morning, one and all. As my topic is design of experiment and optimization in a tool in formulation development. So far, I mean, the previous speakers discussed about the various novel drug delivery approaches for targeting the cancer cells. But my topic is not directly related to the cancer drug delivery, but it is a kind of strategy to improve the drug delivery. I mean, improve the formulation development by using this, novel tool, I can say. Usually, we will do the experiment in traditional way is by keeping, one factor as a changeable one, and all other factors we will keep it as a constant. That's what usually we will do the experiment, any experiment. But to know the effect of that factor, we have to run so many experiments. Every time we have to change a factor and its levels, low level, high level, I mean its concentration. For example, I'm telling a simple formulation. Since I know that most of the the audience are undergraduate students, so for understanding in a better manner, I can say even cold cream, you take as an example, it contains, number of not cold cream. You can take one tablet, any tablet, paracetamol tablet or something. Apart from the API, that is paracetamol, a lot of other excipients are present in the formulation. But each excipients play a major role in the, efficacy of the paracetamol. For example, disintegrating agents, what we are using, or binder, what we are using, also plays a major role with the response of dissolution. You all know that the dissolution is mimicking the bioavailability of the drug. The better the dissolution, better will be the bioavailability. So these factors, disintegrating agent, even sometimes diluent also, even sometimes glide and lubricant, whatever the ingredients used, have some effect on the response. So our response is dissolution. So it all have some effect on dissolution. Sometimes it has some cumulative effect. Lubricant and diluent will have some interaction, and if it will affect the dissolution. For example, if you use both lipophilic glidant or lipophilic lubricant, if you have taken, means that will reduce the dissolution. So these kind of things cannot be performed in a traditional method. So in traditional method, if you do all kind of these studies, means it will take number of experiments, and it will take long time. We have to use more chemicals, more manpower is required. So in order to avoid all those things, the modern method or a modern tool is available to optimize the formulation, is called design of experiment. So with this experiment, tool, we can at a time, we can study the several factors and its response. So in a simple experiment, for example, I told glidant, how it will affect lubricant, how it will disintegrating agent, how it will affect binder, how it will affect on dissolution. In one single experiment itself, we can understand. Okay, so that's the advantage of this particular tool. So in my presentation, I'm going to give some introduction, small introduction about what is DOE exactly, and what are the advantages of DOE over the conventional way of optimization, and what are the various designs used in DOE, namely factorial design, central composite design, box Benham design. And I'm also going to give some case studies especially for central composite design and box Benham design, because these two designs are used by the researcher in common. First of all, you should know what do you mean by optimization. So optimization means to make perfect. That's what. So as a formulation scientist, we want our formulation supposed to be perfect one, perfect in terms of our goal. Our goal is it should have good dissolution, or if I'm making some nanoparticles, means my particle size should be this size, or my entrapment efficiency should be within this range, or my polydispersity index should be within this range. So it is not possible with the traditional way very easily, but by optimizing the tool like DOE, it is very possible to make a perfect formulation. So in a simple way, we can say optimization is the process of finding the best way of using the existing resources while taking into account all the factors that influences decisions in any experiments. That's what. So we no need to go with any other new excipients or something. Whatever the excipients already available are used in the formulation, in that itself we will come to understand that which factor is more influence factor on our response. I told example, dissolution is our response. So among the different factors, I told what is the most affecting or influencing factor, that we will come to know, and accordingly we can decide our experiment. So that's why this optimization is used in pharmacy relative to mainly formulation and processing. So as I told, traditionally optimization is carried out by changing one factor at a time. That is called OFAT. One factor. Every time we can change only one factor, remaining all other factors, keep it constant, and we will change, check the response and evaluating the results. And this process is repeated again and again until obtaining a desired results. So this traditional approach has been proved to be not only too expensive in terms of time, money, and effort, but also unfavorable to fix errors. Yeah, that's what I told already, all those things, but not even we cannot fix the errors of our formulation with this approach. And it is unpredictable. The results are unpredictable, and even sometimes it is unsuccessful approach. Coming to the modern methods of optimization, which includes, just a minute, yeah, which includes the use of systemic tool like the design of experiment in order to enhance the outcomes. So we should know what you mean by DOE. DOE is an organized approach to determine the relationship between independent product variables and their effects on the response variable of the product or process. So just it's an organized or systematic approach to understand the relations between the independent variables. I told you, the tablet example. In that tablet example, apart from the API, all other, ingredients, excipients, what we are used are called independent variables. What is the, effect on dependent variable? Our dependent variable is dissolution, or otherwise called response. That easily we can understand by using this approach. The optimization using DOE helps in reducing the cost, saving the time, preventing errors, and helps in reproducibility. Every time you will get the same kind of results, but that cannot be achieved through the traditional method of optimization. Yeah, this is again coming to the advantages of DOE over OFAT. Yeah, OFAT means one factor at the time. Remaining factors are one factor we are changing, remaining factor are constant. More number of trials or runs are required to know the effect of the particular factor towards the response. But in DOE, only less number of trials or runs are required to identify the factor which is influencing more on the response. Then second point is the number of runs depends on the output of the product or process. Yeah, if at all, if your output is very tedious one, means the number is more in OFAT, but in DOE, fixed number of runs based on the design. I mean, here the design what you are choosing will decide the run. If you choose factorial design, you will have some run. If you choose a central composite design, you have some other number of runs. If you choose a box bend hand design, you have some other runs. So OFAT is a classical type of optimization technique. That means a study of one factor or one variable at time, whereas DOE is a scientific or systematic approach. Here we can study and correlate all factors and responses at a time. Even in single experiment or a single approach, we can understand all the factors influence on responses. In OFAT, consideration of all factors are equal. Okay, we have to consider all factors are equal, so we have to change every time one factor. But here, relative significance of each factors, easily we can identify which factor is more significant, which factors will more influence on the response. That easily identified in DOE. In OFAT, no interaction will be known. That means, what is the effect of combination of disintegrant, lubricant, and binder on dissolution. That you cannot understand through OFAT optimization. But, in DOE, the interactions will be studied. So as such, the lubricant, how it will affect the dissolution in combination with binder, what is the response towards the dissolution. So that is called interaction. So interaction of the lubricant and binder on dissolution will also be studied. And in OFAT, if any experimental fails, rest will be unassumed. Yeah, that's all. But here, DOE, it is a orthogonal design. It's very predictive and very decisive, and now less knowledge from the experiment. So in conventional method of optimization, you cannot, get more knowledge about your experiment. Only one factor you are going to use, so that factor influence on that means that's all. But you cannot, have combination of the factors effect on response, all those things. So here, more knowledge from the experiments, and maximum resources and time is required in OFAT. Minimum resource and time is required in DOE, and you may get inaccurate information from the OFAT, but you will get accurate information from the DOE. Sometimes the OFAT may mislead in the results, but the results predicted by the DOE is always accurate. And sometimes the product subjected to failures also, but here there is very less chance, or no chance of product failure, because these processes are more robust, and the product also will be more robust. It will give always the same results. The reproducibility also possible in DOE. I'm sorry. What happened? Just a minute. Yeah. So the next one is, what are the common terms used in DOE? So mainly these terms are very common. Okay, one is factor, especially in the independent factor. Okay, another one is response or effect, then levels, then center points. So these are the common terms generally used in DOE. So first, let us see what is factors. Factors are otherwise called independent variables. I told you, in the same example, you can take tablet, this binders concentration, disintegrants concentration, or lubricants concentration. These are all called independent variables. And it's also called control factors. We can control. We want to use that factor, we can use. We want that variable in this much concentration, that is possible. So that's why we can control, but it may or may not depend on each other factor. Sometimes it have an interaction with other factor. Sometimes it may not have an interaction. Sometimes, this binder and the lubricant will increases the dissolution. That means there is an interaction. Sometimes the binder and the will not influence, or there is no interaction with the lubricant. As such, if you change the binder concentration, some differences will be there. But there is there is no interaction with lubricant. It is called input variables, which can be changed. Okay, that's what I told, excipients and concentrations. So we can what is called change the concentration. Okay, these are the input variables. So independent variables are otherwise called input variables or controlled factors. Then response. If you give input, automatically there is output. That output is nothing but the response, or otherwise called the effect. They are called dependent variables because it has to depend on independent variables. See, if you increase the disintegrants concentration, obviously we know that the dissolution will be increased. Or if you increase the binders concentration due to hardness, the dissolution will be decreased. So that's what the effect is, mainly depends upon the independent factors or independent variables. So that is response or effect. So it is a measured output value. We can understand this much percentage will be the resolution or something. Then levels. It is also a specific value of the factors. Again, this is also input. So we have taken disintegrants concentration in two different concentration. One as a low concentration, one as high concentration. What will be the effect? That is called levels. Usually it will be more than 2 because obviously it should be, one should be low, one should be high. So these high and low levels can be generically coded as +1 and -1 in DOE. For example, a two-factor experiment. Two-factor means definitely it will have two levels. So it is called two-square factorial design. 2 into 2, 4 experimental runs will come. And center point. Center point is the center value of all factor ranges. Anyway, in the next slides, you will understand the role of center points. Yeah, again, I want to give some more example for the same common terms. Factors, already I told, it's an assigned variables such as concentration, or it may be a temperature, whatever. There are two types of factors. One is quantitative factor. Here, the numerical factor is assigned to it. For example, concentration, 1%, 2%, 3%. Another one, qualitative, but they are not numerical. Example, the polymer grade, humidity conditions also we can give. Yeah, that's what on the side you can see. The factors may be a temperature. Temperature is, I think, it's a qualitative factor. We cannot quantify. And the numerical factor, concentration like 1% or 2%. And the levels, one is low level, one is high level, you can able to see. So levels is a factors or the values, or we can say it's a designation assigned to the factor. I mean, what is the concentration, what is the temperature, all those things. Response, it's an outcome of the experiment. Already we discussed, is the effect to evaluate the factors. So, for example, disintegration time, all those things are the dissolution, also the response. Effect means it's a change in response caused by varying the levels. So when you will have a proper effect, if you change the levels of the independent factors, there will be a change in the effect. So it gives the relationship between various factors and its levels. Okay. Example, increase in hardness of the tablet is the effect of binder concentration. If you increase the binder concentration, obviously there will be a hardness of the tablet will increase. That's the effect of binder's concentration on tablet. So interaction, it gives overall effect of two or more variables. That's what I told. It's a combination of two more variables effect on the response. So for example, combined effect of lubricant and glidant on the harness of tablet. Hope you, I mean, with this slide, you understand some of the common terms. Now let us move on to the next slide, which describe what is experiment. Since this tool is named as design of experiment, mainly what we are using this tool to design the experiment, to optimize the experiment. So we should know what is meant by experiment in a expanded manner. So in statistics, an experiment refers to any process that generates a set of data. If you get some data from any process, that is called experiment as per statistics. Or we can say an experiment involves a test. Yeah, usually experiment is a test or series of tests in which purposeful changes are made into the input variables of a process system so that the changes in the output responses can be observed or identified. That's what that's a easy example to understand. Experiment is just I'm changing the concentration of disintegrants in a tablet formulation. I will accordingly get the results in dissolution. Okay, so that's an experiment. So next we can say experiment is determining which variable, that means n input, we can say x, that is independent variable, which is most influence on the response, that is output, we can say y. So that is also experiment. So which factor will have more influence on the response, that is also experiment. If you determine means, if you determine where to set the influence x so that y is near to the nominal requirement. So in which concentration of the factor, if you set means we can get the expected result. That also a experiment. Then determining where to set the influential X to the, so that the variability is Y is small. Suppose Y is your, error or the problem of your response is the problem of your product or the formulation. So you want to reduce that error, your Y value, where you have to, set your X factor. That also we can determine through experiment. So determining where to set the influential X's so that the effects of uncontrollable variables also Z also minimized. In experiment, not only there is a factor, there is a response in that happen in the process. So apart from these, controllable input factors in the process will give always response or output measures, we know very well. But apart from controllable factors, some uncontrollable input factors also will be involved in the process, which may affect the response. So we have to know something about uncontrollable variables and how we can minimize the uncontrollable variables by setting the controllable input factors. That also we should know. So what is controllable input factors or X factors? These are the input parameters which can modify in an experiment or process. So controllable input factors or X factors are independent variables which is modifying the experiment. For example, in cooking rice, the factors includes quantity, quality of the rice, and quantity of water used for boiling is the controllable input. So it, this will, modify the, if you change the rice, the process will change. Or if you, reduce the quantity of water for boiling, the boiling process will not be okay. So these are all controllable input. So what is uncontrollable input factors? Are those parameters which cannot be changed. So here you cannot change, because in the same example, rice cooking example, this may be the temperature in the kitchen. Yeah, you have a boiling, you have a rice, quality of rice, you have a quantity of rice, you have a quantity of water for boiling, and you are started, boiling. But in the temperature in the room will also influence the your boiling effect of it. If the temperature, if it is in summer, it will boil quickly. If it is a winter, it will boil, little delay. So that temperature is called uncontrollable input factors that also, influence the response. So that factor also need to be recognized in order to understand how they also will affect the response. So response or output measures already we discussed, but still these are the elements of the process outcome that gauge the desired effect. In cooking example, the response may be the taste or the texture of the rice or the response. And interaction, when an experiment has 3 or more variables. So in the same example, how many variables are there? Cooking rice, one is quantity of the rice, another one is quality of the rice, the third one is the quantity of water. So 3 variables. So the 3 variables in an interaction is a situation which is the simultaneous influence of 2 variables on third is not a additive. So maybe the quantity of quantity of the rice and quality of the rice will influence on the effect or response rather than the quantity of water what we are used. Or sometimes the quantity of rice and the quantity of water used will have some effect on the rice boiling. Okay, so this slide I especially kept to understand what is controllable factors, or otherwise called independent, independent variables. And uncontrollable input factors means what? How it should be identified, and it should be how it should be affect the response also we will let us know. And what is response and what is interaction, so far we learned. And what are the various steps involved in DOE? These are the steps. First, we have to set the objective. Objective is, for example, I'm formulating a nanoparticles. So I am setting my nanoparticle size should be within this range. That is my objective. Then I have to set the process variables. So mainly in nanoparticles, we know that, for example, lipid-based nanoparticles means the lipid concentration will affect the particle size, and the stabilizer or surfactant what I am using in the lipid nanoparticles will affect that. So I have to set the process variable. So one is lipid concentration, another one is my surfactant concentration. Third step is selection of experimental design. Then, so I have only chosen 2 factors. Now I want one more factor, or I want, one more factor is my, the charge inducer used in the nano formulation also will influence on some effects. Means based on the factors. So if it may be a 2 factor, it may be a 3 factor, and based on that factor, you have to choose the experimental design, whether it should be a factorial design or central box composite design or box-Benham design. And then you have to execute the design, run the design in the software. Then check the consistency of the design suitability to variables, whether it is a, validated one or not validated one, you have to check. And then the last 6th step is analyze and interpret the results. Then final step, you will get the optimized formula. You will extract the optimized formula. This is what, in this manner, I will show some experiments as a case study after some time. So then what are the different types of DOE? As I told, one is factorial designs, another one is response surface methods. So again, factorial designs are classified into full factorial designs, fractional factorial design, and miscellaneous factorial. So under miscellaneous factorial design, blocked Berman design is there, Takuchi OA design is there. But blocked Berman and Takuchi is not much used nowadays because of its, improper results, or it's not very user friendly. And response surface method is the most common method used by the researchers for optimizing their formulation. So the under response surface method, we have 2 methods, namely box-Benham design or method, PB design, and another one is called CCD or central composite design. So these 2 designs are used. In full factorial designs, generally we will go up to 5 factors. If you have more than 5 factors in 2 levels, then it will have more number of runs. So in order to avoid the number of runs, we have to minus the insignificant factors through fractional factorial designs. Similarly, in central composite, what is the difference between central composite design and box-Benham design means? The box-Benham, if you know the lower limit and upper limit of your variables, then better you can go with composite, central composite designs, which will always, optimize with less runs. But box-Benham design is the advantage over sometimes, you can go with box-Benham design. If you have, if you don't know the upper limit and lower limit of your factors, independent variables means, you have to go with central composite design, because in central composite design, we will have star points. The star points will cover a little above the range or level of our concentration. Yeah, user guide. So, sir, you told so many designs. So how I have to choose the right design for my study means, this guide will be very much useful. If you suppose if your factor is only 1 factor you want to study means, you don't need to go with any optimization design. Just it's a 1 factor. Completely, it's a randomized design. So there is no screening objective. You don't need which factor is having influence on my. That is called screening objective, or response surface objective. Also, you don't have. Suppose you have 2 to 4 factors you are using in your experiment, then you have to use full or fractional factorial design, if at all screening objective. What is screening objective? Screening objective means just to know the influence of your factor on response. That's all. But at what extent, or if you change the concentration less or more, what will be the effect, or what is the combination of my factors on the response? That and all you cannot, screen from the full factorial or fractional factorial design. But whereas in central composite or Boxman and design, you can do all those things. I mean, you not only you can screen, you can also understand the cumulative effect of your factors on the response, or even interaction studies, as I discussed in the earlier slide. The interaction effect on your response also can be studied from response surface objective. Then if you have more than 5 factors you are supposed to use in your formulation or experiment means, then that is called again randomized block design. The comparative objective is screening objective. You can use fractional factorial or Blackett-Berman. Here you cannot use full factorial. As I told you, what is the difference between full factorial? In full factorial, you cannot go more than 5 factors. If the factors are more than 5, it's better to go with fractional factorial design. That's nothing but it's a kind of full factorial designs which minus the insignificant factors which influence the response. And yeah, that's what, even in response surface method also you can go. So screen first and reduce the number of factors which are insignificant on your with respect to your response or effect. So now let us have some idea on factorial designs. What is factorial design? So this design is mainly used to screen the major factors that affects the product. Okay, so I told the first one example, tablet formulation. So tablets can, apart from the API, there are many excipients. All the excipients we are considering as factors. So among the different excipients, which excipients mainly influence on dissolution, that we can come out with this factorial design. So factorial design allows for the simultaneous study of the effects that several factors may have on the process. That's the beauty of this tool. At a time, you can understand the effect of or influence of several factors on response. When performing an experiment, varying the levels of factors simultaneously rather than one time. So in one, usually in conventional method, one time we are changing the factor, we are changing the levels, something. No, here in 2 levels, all the factors we can change, and it is very efficient in terms of time and cost, and allows for the study of interaction between the factors. Yeah, interaction study also we will get. Main effect also we will understand of individual factor, and interactive effect of the combination of factors also we will understand through factorial designs. The number of ranks can be calculated by the formula levels to the whole power factors. Okay, so usually factors can be denoted by the letter K. Okay. So as I told, factorial designs are classified into full factorial and fractional factorial. Again, full factorial is classified into 2K factorial designs and 3K factorial designs. But usually in full factorial, people generally use 2K factorial rather than 3K factorial, because 3K factorial will have more number of ranks, which is not solve the purpose of our using DOE. So in full factorial experiment, the response are measured at all combination of the factors levels, which may result in a prohibitive number of ranks. For example, a 2-level factorial designs with 6 factors requires how many ranks? 2 to the power 6. So 2 into 2 into 2 into 2 into 2. So 64 ranks. A design with 9 factors requires 512 ranks. But these many experiments we cannot perform. So in order to minimize the time and cost, you can use the designs that exclude some of the factor level combinations. So in this 6 factors or 9 factors, some factor combinations, 3 factors or 5 factors, we can omit. That is called, fractional. So factorial designs in which one or more level of combinations are excluded are called fractional factorial designs. So this is the number of factors. If you have two factors and two levels you have to study, high level and low level. So 4 runs you require. 3 factors means 8. 4 factors means 16. So till 16, I think it is possible. So if you have more than 5, it's better you can go with central CCD or BB design, which will have less numbers. Okay. And here on the right hand side, you can understand the factors of A and B. Factor A is the horizontal one, and factor A B is the in x-axis, and factor B is in y-axis. So now I will put a center point on this particular. Just a minute. Yeah. So now here this will be the center point. Then I will divide into 4 columns. Then I will get these many levels. Okay. In the first point, I'm having A, high level, and B is the low level. Similarly, 2, 3, 4 also you will get. These are the levels. So totally of 8 runs I have to make in case of 2 to the 2 to the power 4. Sorry, 4 runs I have to make. 2 to the power 2 means. I mean 2 levels in 2 factors I am using means. Similarly, I am using 3 factors. 2 to the power 3 means I have to. I will run 8 factors. And similarly here also, if I will put the center point here, it is a cubical one. It's a cubical one. 3 factors means. So factor A is on x-axis, and factor x-axis factor B, y-axis factor A, and the cubical this will be the C. And if I will put a center point here, I will, and then if I divide into 8, and 8 points will have one in point 1. A will be the high level, whereas B and C will be the low level. Similarly, these, all the levels will come. Okay, just for understanding. Next one is response surface method. So RSM is a collection of mathematical and statistical technique useful for modeling and analysis of problems. Whereas in factorial designs and, we cannot analyze the problems, where in response surface method, we can analyze the problem where a response is of interest is influenced by several variables, and the objective is to optimize the response. Okay, so it defines the effect of the independent variables and interaction of the variables on the process. So what is the effect of variables and what is the interaction of variables? That is not possible in full factorial designs. It generates mathematical modeling which describes the process. So the surface response surface is visualized by response surface and contour plot. So these are the methods used in surface response surface methods. One is central composite design, and another one is box Benham design. So now let us see something about central composite design. So central composite design contains an embedded factorial design. So it is nothing but a factorial design embedded with some points. That points are called star points. It allows the estimation of curvature. This allows the finding the interaction of the excipients with the results out of the limits also. The star points are represented by the symbol alpha. The minimum number of factors that can be taken, that's one condition. For central composite design, minimum 2 factors should be there. And it is also like a factorial design. CCD includes the run with all the corner points in addition to the star points and center points. Usually in factorial designs, we will calculate only the corner points, but in CCD, we will also calculate or consider the star points. So this is the normal factorial design. So I put in the previous slide. No, this is the A factor and this is the B factor. So if I will put center point here, I will divide means I will get 4 points. So so this corner points only we will generally consider in case of factorial design. but in CCD, this is the extra. So this is called, this alpha is called star points. So just I will impose this axial points or the star points to the 4Q points, means I will get like this. So apart from this corner points, I will have extra star points. So this will covers the, apart from the limit. So that's why I told this design is more suitable. If we don't know what is the upper limit and lower limit, means I can use this central composite design. Okay, so this is the 2 factorial central composite design, and this is the 3 factorial central composite design. Okay, so these are the Q points, and this red color is the center point, and the yellow color denotes the star points. Now let us move on to the case study. Okay, so this will be very much interesting and will be useful for you if you have the tool, whether it may be a free trial tool. If you have a free trial tool or licensed tool, you can try how I am following. The similar way if you follow, easily you can optimize your formulation. It's not so, tough or difficult to run, use this tool. Okay, so for example, one of my students, I'm not going to discuss everything. He has worked on the formulation and optimization of tolfenamic acid loaded nanolipid carriers for colon cancer using central composite design. This is his title. So based on his formulation is NLC, nanolipid carriers, nanostructure lipid carriers. So he used both solid lipid and liquid lipid, and he has taken this combination. One in upper level, he put 2% and lower level 0.5% he has taken. And the second factor he has taken is twin 80. That's a surfactant or stabilizer used in the nano formulation. Again, he has taken one high level 1.5 and low level 4. And what are the response he is expecting on these 2 factors? How it will respond to particle size, polydispersity, entrapment efficiency. So this is what you want to know. What is the effect of these responses? These factors on these responses. That is his optimization. Okay, now let us move on to the tool. Okay, so this is the first. If you have a software, if you open the software, this page will come. Okay, whether even if you try trial version, which may be sometimes available in the online. So if you open, then if you just click new design, or if already you have stored something, means then open design. Otherwise you can new design, because already I have loaded the values and everything just to give the demo. These are the already stored files are available here. So I have to open the design. If I will click open design or new design, this page will come. Okay, so in this page there is a method called response surface. Okay, response surface. So in the response surface, central composite, box pen hand, all those designs will be there. So I have to click central composite. If I will click central composite, then it will ask how many factors you want to use. I have to put here 2 factors. Automatically it will take one is low and one high, all those things. And totally 2 factors means there will be 8 corner points, non-center points, and there will be 1 center point will be there. I will show you here. Yeah. So 2 factorial design, composite design I have taken means this will be the central points, 1 center point, and there is around 8 corner point or non-center point will be there. Okay, so this and all automatically will come. Only thing is you just go to the response surface, then click central composite, that numerical factors. Just just a minute. I want to put this down. Yeah. So the first step, open the design expert software. Step 2, response surface. select CCD design and select the factors and enter the level. Okay, so 2 factors I have selected, and level, one is low and one is high. Automatically it will come. Then I will go to the next. I have to click next. Then this box will come. I mean, so here, how many response you want to study? You want to study polydispersion index, particle size, entrapment efficiency. So for example, here I want these 2. I will put 2 and I entered. One is name of the response is PDI, polydispersion index. Another one is entrapment efficiency. Then I have to click finish. Then this slide will come. If I finish means it will give total of 9 runs. I told you, 1 center point and 8 non-center point. Total 9 run will come. So this I told this is the maximum. I put high level and low level now. So in 9 runs, what are the other possible combinations you can go for? All those things will come, and then now I have to perform this experiment, 9 experiments. Then I have to take the result of particle size for all these 9 formulation I have to enter, and I have to enter the polydispersity index and entrapment efficiency I have to enter. And after entering, just I will click this page, which will go to the next one. Okay, so enter the responses for the runs given by software. The 5th step is the select of the select the response such as particle size. Okay, so now you want to analyze which response you want to know with respect to your factors. Means, for example, here these are the analysis. There is one column called analysis. So here particle size PDA. So I want now analyze only the particle size. So I have to select this particle size, and then I have to click. Then I will get this summary. So after analyzing, check for the fit summary, ANOVA, model graphs, and all those things will come. So here, from this, what I understood is this design is useful design, and there is a influence of our variables on response, and I can go further. That's why there is a response suggested. Okay, that means I can go with further, like. Okay, so after this, again I have to go to the next step. I mean, next steps in the same, everything will be all information will be in this side. Okay, so if I will go to the next step, so analysis, particle size analyzed, and again here model graphs. So by clicking this model graphs option, I can get various kind of graphs such as perturbation, contour plot, 3D surface plot. So now I will click perturbation. So I got the perturbation plot. So perturbation plot. So here the center point is the response, and B is the one factor. I think what I entered in the beginning. A is the another factor. So the B factor is not deviated the center point much, but whereas A is deviated much. So from this, what I understood is the factor A influencing my response much. That's what. In the perturbation plot, the lines indicate the factors. The higher the deviation of the line from the center dot, which indicates higher influence of the factor on the response. So here, which factor is influenced more? Factor A influence more because it is deviated more when compared to B. Okay, so that I understand. Now, next slide. I mean, next step is same thing, the particle analysis. If you select PS analysis and model graphs, now instead of perturbation, I clicked 3D surface. If I will give 3D surface means, then I will get this particular picture, 3D picture. So here, this is the factor B, and this is the factor A. Factor B is my surfactant, factor B is the lipid ratios. So if I will increase the surfactant, this is my response. It's not increasing much. It is not, it's increasing, but not significantly. It's increasing very slightly. But when I increasing the lipid concentration, you see the response, it is also, it's increasing much. So from this, easily we can understand. So the perturbation graph, what we got, and this also same results. B is here also, B is, influencing more. Here, the particle size is increasing like anything. Okay, when we increase the concentration of lipids, the particle size also will increase. But when we increase the concentration of the surfactant, the particle size is not increasing. Okay, that you can understand from 3D graph. Now, next, same, you are in the same page, analysis and, particle size analysis. Now, you just, instead of so you understand the effect of particle size on your response. Now select the numerical option from the optimization. You can, now we are going to, derive the, optimized formula. So for that, you have to go to the optimization and select numerical. Okay. And clicking, then, after selecting the numerical, you just click this criteria. Okay. If you click the criteria, then which criteria you want to analyze. So as I already, we know that, we are analyzing particle size. So we have to take particle size. And what is your goal? So particle size, you know, in the nanoparticles, always it should be minimized. Actually, by here, I put none. Actually, there are three options will be there: maximize, minimize, none. So actually, our goal is one is to increase, or another one is to decrease. In this case, in nanoparticles, our goal is we have to decrease as the particle size. I'm supposed to select minimize, but it is not, I didn't do it just to understand. There are three options there. In the next example, I have selected the minimum. Okay. So here the formulation is nanoparticle. So the particle size should be set for minimum size rather than none. Actually, here none I am not supposed to select, minimize. And even in same case for polyspecificity also. But in case of entrapment efficiency, should be maximum. So I have to select maximum here. Okay. Then next slide. Yeah. Optimization, numerical. Instead of criteria, now I have to select solution, and then I have to select ramps. Okay. I have to select ramps to get my optimized formula. So after selecting, I get, this is my optimized formula. So I have to select this A factor, this, and B factor. This means I will get the particle size of this. I will get a polydispersity index of this, and entrapment efficiency of this. That's what. Then after getting this formula, once again I will do the experiment and I will confirm the results. That is called validation. So for the given criteria, the optimized formula of factor A, that is solid and lipid liquid, then if taken at 1.65 and factor B at 1.5, the nanoparticles with the minimum size I will get. That's our goal. No, our goal is, this is the minimum size which is possible, and this is the minimum PDI, and with a greater entrapment efficiency I will get. Okay, if you are okay with this, you can just go ahead for the experiment, and then you can see the observed value with your predicted value. Both the values are close, then your design is validated. Hope you understand. Okay, so with this I'm concluding. Sorry, one more features also I'm having. So the optimization, then instead of numerical, now I'm selecting graphical, and then I will put graph this overlay plots. Okay, so by selecting graphical option from optimization, I can get graph option, and we can get the overlay plot. This is called design space. Okay, so here we have all the 9 points. Even if you use this 9 in this point, what is the particle size, what is the factor A concentration, what is factor B concentration, what is your entrapment efficiency, everything. Even in this design points, you can, any point you can click, that point will give what is the concentration of your factor A and factor B, and with respect to its particle size, entrapment efficiency. This is another advantage. So if you want to reduce or more, you just click anywhere, you can accordingly change your formula. So for that, this is useful. Okay, overlay plot or design space plot. So the ideal conditions. Hope I have another 5 minutes to 7 minutes. Excuse me.
Yes, sir, you can take 10 minutes. No issues.
Yeah.
Thank
So ideal conditions for CCD, if there are only two factors, the optimization can be done by using central composite design. In case of box penhand and CCD, both are categorized under same optimization design. Only CCD is the option as BB needs a minimum of 3 factors. That's what. If you have only 2 factors, that time you want to go central composite design or BB means you have to go with CCD because for BB, you need 3 factors. If there is no confidence about your upper level and lower level of the factor, CCD is the best design for optimization, as I told earlier, as it involves the study related to extremes. So below and lower levels of the given levels by means of star points. So this includes the run covered by the simple factorial design and additional star points runs, thus giving an efficient outcome. Now let us move on to the box Benham design. So box Benham design is another optimization design, which is mainly depends upon the middle levels of all the 3 factors. So in all other designs, we have only high level and low level. But in box Benham design, we have one more level called mid level. That is, medium level. Apart from high, lower, medium level also will be considered. That will be you mainly optimize your box Benham design. So the influence of the excipient in the middle level range, which reduces the number of runs and thus makes the optimization easy. So the minimum number of factors need to assign the box Benham design is only 3. The design avoids all the corner points and star points, which is available in central composite design. So automatically there is no inclusion of extreme levels. So automatically the number of runs will be reduced. So number of runs will be calculated in box Benham design with this formula 2k into k minus 1 plus number of center points. For example, number of factor k are 3 and number of center point 1 if you take means 2 into 3 into 3 minus 1 plus 1, you will get only 13 runs with this formula. And this is the center point, and these are the non-center points. And here there is no star points. In central composite design, there will be a star points, but here there is, because here we know already what is the upper limit and lower limit. Only the medium or mid level will be taken considering with this design. So yeah, I want to discuss another case study for box Benham design. Similar way. So another, for example, our study is formulation and optimization of disulfide loaded solid lipid nanoparticles. So now 3 factors I have taken, because for box Benham minimum 3 factors is required. One is surfactant percentage, lipid quantity, solvent volume I have taken, and these are the levels. Level 1 lower, and medium level also I have taken higher level. So that's a speciality of box Benham. So we have to have medium level also here. So here also the response is particle size and entrapment efficiency. So I move on to the software and I open design or new design. It goes to this first page, and here what I'm doing is, I'm just click the box Benham. In the response surface, I'm clicking the box Benham, and I will put how many factors. In the previous design, we put 2 factors. Here, we have to put 3 factors, and then we have to click next. So this box Benham design is implemented with 3 factors, 5 center points, that will give 17 runs. So here you can see how many center points. Center points are 5 center points are there, and 3 factors are there. So it will give 17 runs. Yeah. So next, in the box Benham design, select the response and name. So my response I want is particle size and entrapment efficiency. Then I will just click finish. Then it gives the formula. It gives how many runs? 17 runs it gives. So these are the combination of the formula. and these are the response. I mean, after making the formulation, I have to go and characterize the particle size and entrapment efficiency, and then I have to enter here, and then I have to go to the next step, analyze. Okay, select the response such as particle size and click the click start analyze. Okay, so in analysis, I am selecting particle size and I am just clicking. Then it will use the fit summary, ANOVA and model graphs. So here also it is suggested. That means our factors are influencing on the response. So we can go ahead. Then I will particle size, I mean analysis, particle size analyzed. I will click model graphs, then perturbation, I will get. So here also 3 factors, So which factor is influencing, deviating more? The factor B in from the center point. So automatically the lines indicates the factors. The higher the deviation of the line from the center dot indicates higher influence on the factor response. So here also B has more influence on particle size than A and C. And similarly, particle size analysis, I have to go take the model graphs and 3D surface. So here also I can, explain you the factor A, B. Yeah, similar. If I will, even C will also will be there. But, factor A is not much influence. So when increasing, the particle size is slightly increasing and then started decreasing. But I will increase the lipid concentration, the particle size will be increasing like anything. So I have to reduce the lipid concentration in order to decrease the value. So next numerical, same, I will select and then particle size is my in the criteria particle size. So here goal is minimize. I told last time I put none. Actually, particle size means we have to put minimize. By entrapment efficiency means because we need more entrapment efficiency, so maximize. Then I will click. Then I will get the optimized formula. So this is a surfactant concentration. This is a lipid concentration. This is the solvent. I have to choose. I mean amount volume I have to choose. So I will get the minimum particle size of 100, maximum entrapment efficiency of 32, and like this kind of details you will get. And similarly, go to the graphical and enter the criteria, and you enter the contour graphs. Then you will get this overlay plot. So overlay plot will give. If I will touch here, or I am touching here, so here you can see what is the particle size, what is the entrapment, how much concentration of factor 1, 2, or 3, whatever. How many factors we have given, and that many factors will come. So that's the advantage of this overlay plot. So ideal conditions for BB design. If there is a perfect knowledge about the upper and lower limit, then BB design can be efficiently used for optimization, as the mid levels are used for optimization. The interaction between the factors on the response can be determined easily and accurately with this design, as the mid levels that are considered in this design are ideal to check the interaction among the factors. Yeah, that's the reason. So why this mid level is very much easily it will it will interpret the interaction between the another 2 levels, high level and low levels. Not only the factors within the level, different levels are there. That levels, how much influence it's on the response will be easily considered or easily judged or established by this mid-level points. So the number of run is less when compared to that of the central composite design, as there won't be any star point runs. Yeah, here since there is no star point, so number of runs will be less. So these are some of the softwares available for design of experiment. The one is the design expert. What I have shown that is design. Apart from that, DOE++ one software is there. Minitab is there. MATLAB is there. So anything you would like to work or adapt, you can adapt, and you can easily what is called optimize your formulation. So coming to the conclusion, so DOE is the systematic and organized tool for optimizing the formulation and determining the relationship between the factors to output of the product. The design software drastically simplifies the previously laborious and hand calculations which needed before the use of computers. So currently DOE software has gained the acceptance as a pivotal development tool in diverse industrial process. Not only in formulation development, its application is wide. Use of DOE as a leading edge approach has become an integral and regular phenomenon in drug delivery. So nowadays, people are using DOE like anything with respect to drug delivery system optimization. So employing DOE optimization can make it much simpler to modify the existing formulations and meet the redefined objectives. So you can change your objectives accordingly. You can optimize your formulation with the help of DOE. However, its enormous potential has not been harvested in drug delivery development, research and industry. We have yet to make the most of this revolutionary practice for optimizing the DDS, drug delivery system. So, so far, in drug delivery system, only in few, optimizing the formula, we are using DOE, and we can explore much more with the DOE with respect to formulation development, as well as in other aspects of drug discovery and drug development process. With this, I would like to, thank the leadership, JSS Academy of Higher Education Research, Mysuru, and JSS College of Pharmacy, Ooty. And I also would like to extend my thanks to the Amity University, especially Amity Institute of Pharmacy, especially the coordinators who are organizing this conference and given me the opportunity to share my knowledge to the pharmacy profession. With this, I would like to conclude my presentation. Thank you one and all for your patient listening. If you have any queries, you can clarify with me. Thank you one and all.
Thank you very much, sir, for the wonderful explanation of a statistical tool for the formulation optimization. We would like to give you a virtual felicitation to our guest speaker, Dr. Natarajan Jawahar, for sharing his expertise in this session. Please give huge round of applause to sir.
Thank you.
Thank you, sir. Dear all, we have had a productive and aspiring time together. As this event is about to come to the end, I hope you have found the sessions at this conference informative and helpful. May I now request Dr. S. Mohana Lakshmi, Director, Amity Institute of Pharmacy, for her concluding and final remarks, vote of thanks for this session. Ma'am, please.
Good afternoon to all of you. So we have come almost to the end of this 2 days conference, and just one session, scientific session, is yet to be done. That will be done in the afternoon. So I first of all thank the organizing team for giving me this wonderful task, proposing vote of thanks. So it is always very pleasant and happy feeling to say thanks for any favor that we receive, right? So similarly, the same feeling that I have now while I am trying to propose this vote of thanks. So first of all, I would like to thank a very charismatic, eminent, dynamic leader, our Honorable Vice Chancellor. We are really very grateful to work under his leadership. His motivation and support always helped us to conduct these kind of programs for giving knowledge to the students beyond the curriculum. So I thank again to our Honorable Vice Chancellor for giving us this opportunity. Second, I thank our Pro Vice Chancellor, Professor MP Kaushik, who is always with us while conducting these kind of programs. Not only for programs, he always holds our hand, especially during unfavorable situations in crisis and challenges, and see that we don't succumb to such situations and we always, stand mighty. So not only for this, for every activities of EIP, his support is very, very crucial. I thank him for this. And coming to all the speakers, guest speakers who have done yesterday and today. So yesterday, Dr. Anup, I just want to say a few words about Dr. Anup. Dr. Anup is not only a keynote speaker for this conference, he is also a well-wisher for EIP. He is a good collaborator, research collaborator and academic collaborator for us. And he works very closely along with our team. And he is very humble and very sincere, which makes him more great. So I sincerely thank him for his wonderful and amazing presentation yesterday he had done and for all his support to AIP. Yesterday he had presented on cost analysis. I hope you all might have listened and benefited out of it. So how we can, analyze the cost of novel drug delivery and conventional drug delivery. He presented in a very elaborative manner with case studies and examples. So second, Dr. Sharath, yesterday he presented all the novel drug delivery systems that are available for, delivery. He started with neosomes, liposomes, he up to the dendrimers. He presented in a very elaborative way with examples and case studies. I thank him for, spending his valuable time with us yesterday. And today we had two great speakers, Dr. Gautam and now Dr. Nadarajan. So both of them have done a very wonderful job. Dr. Gautam in his nanofibers complete starting from the formulation and analysis and evaluation, and now Dr. Nadarajan on the design of experiment with case studies and examples and softwares. Really, guys, you don't know, even though a person is expertise in a field, a person has 100% of knowledge in a particular field, to deliver for 1 hour, he need to put a lot of effort. He has to work at least for 2 to 3 days to make his PPTs and to, prepare himself to make how we can present interestingly. So they have done a wonderful job. See, this will be done not by all. All will not choose to disseminate their knowledge, what they have gained. Only few people have interest in doing this. So the 4 keynote speakers, the resource persons we are fortunate to have for this conference is really a very great thing for us. And I am always grateful for them. I thank profusely for them for their effort. Next, coming to the organizing team. The organizing secretary, Dr. Neeraj, he has done a very wonderful job. The Department of Pharmaceutical faculty members, Dr. Jovita, Dr. Shilpi, Ms. Anita, Ms. Ankita, and Dr. Pratap. The way they divided the task among themselves, the way they meticulously planned and executed. This conference is very amazing. I'm very happy to be backed up by this team, and the leadership that is provided by Dr. Neeraj is very amazing. I appreciate and congratulate them for this conference. Next one is the scientific team. Dr. Bhatt, Dr. Shilpi again, Dr. Satish, and who else? Dr. Raula have done a very wonderful job. We had 400 registrations for this conference who attended both offline and online and get benefit out of this conference and presentations. And nearly 100 papers we received, out of which 60 has been shortlisted by our scientific committee and 30 has been presented yesterday and 30 will be presented in the afternoon. So this would have not been possible by the tremendous effort that this scientific committee members have rendered. I thank them and I congratulate them and I appreciate them for taking their time and doing this job. And next one is the disciplinary committee, Dr. Naveen Sharma. He had done a wonderful job in maintaining discipline. You all know that. He maintained the utmost discipline in the seminar hall, and all the non-teaching faculty and all the rest of the team, whom I have not mentioned the name, Anita and Deepak and Vijay Raj, Murugesh, all have done a wonderful job. I appreciate all of you for your effort. Our IT team. Always our IT team is known for doing a very excellent job, and today is not an exceptional. Mr. Vijay, he was with us throughout the program, even before the program, under the leadership of Mr. Ram Tamar and the central IT team, Maqsood. I thank these three people profusely for helping us. See, conducting a program in hybrid mode, is very difficult compared to doing it in either online mode or in the offline mode. It is a combination of these two. So we have to, see whether these two properly hyphenated. Otherwise, it will create chaos. So they have really done a very wonderful job. Again, I thank Mr. Vijay for this. So last, students, there is a saying, we can only lead the horse to the water. It is up to the horse whether to drink the water or not. Am I right? So now we have successfully led you to the water. How much of the water you have consumed? You all know that, whether you have consumed 1 milliliter or 1 liter or 5 liter. See, if the horse is adamant in not consuming any water, who is going to be the loser? The person who is leading the horse is not going to be the loser. The loser is the horse. The horse is only going to be dehydrated and the horse is only going to be succumbed. So my dear students, I sincerely believe even now, you might have benefited definitely out of this conference and also with our future programs. So be open-minded and open-hearted to receive anything in a positive manner. So I thank once again to all of you for taking the time to choosing to spend your valuable time in the seminar hall for this 2 days. I also want you to attend the afternoon scientific session and get benefit out of it. And all the best for your presentations in the afternoon. Thank you all of you. Best of luck.
Thank you so much, ma'am. Now I would like to request all the online participants to fill the feedback link that have already been shared in the chat box. And the link will be same for generating your certificates. And certificates for oral presentation for all the online participants will be sent to you within 1 week. And the next one is the scientific session for the delegates who have submitted their abstract for oral presentation will be held from 2 p.m. onwards in the seminar hall block C. I request all the participants to attend the session on time for a smooth conduct. Thank you all. Thank you, IT team. Sir, can we close the session?